Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Mutations and DNA damage explained. Table of content. Introduction to mutations and DNA damage. Importance of mutations. Classification of mutations. The molecular basis of mutation. General classes of DNA damage. Introduction to mutations and DNA damage. A mutation refers to any change in the genetic material that is heritable, meaning it can be passed on to the next generation. Mutations can be induced through exposure to radiation as well as various chemical agents. These agents are capable of causing strand breakage in DNA. They may also produce other types of damage to DNA that can alter its structure and function. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Importance of mutations. Mutations hold fundamental importance in molecular biology because they serve as a major source of genetic variation, which is essential for driving evolutionary change. Mutations can result in harmful, deleterious effects, though in rare cases they may provide advantageous consequences to an organism or its descendants. When mutations occur in germ cells, they can lead to heritable genetic disorders. In contrast, mutations in somatic cells may cause acquired diseases such as cancer or various neurodegenerative disorders. Mutant organisms are valuable research tools for molecular biologists, as they help in the characterization and understanding of genes involved in different cellular processes. Classification of mutations. Mutations can be classified in several different ways. In multicellular organisms, one important distinction is based on the type of cell in which the mutation first arises. Mutations that occur in cells destined to form gametes are known as germ-line mutations. In the case of somatic mutations, reproductive cells remain unaffected, meaning that the mutant allele will not be passed on to the progeny. In higher plants, somatic mutations can often be propagated through vegetative means, such as grafting or rooting of stem cuttings. Nucleotide substitution is a mutation in which a nucleotide pair within a DNA duplex is replaced by a different nucleotide pair. Mutations that alter a single nucleotide pair are specifically referred to as point mutations. Certain mutations cause more extensive or drastic alterations in DNA, including 1. Expansions of trinucleotide repeats. 2. Extensive insertions and deletions. 3. Chromosomal rearrangements. 4. Insertion of a transposable DNA element. 5. Errors in cellular recombination processes. Conditional mutations are mutations whose effects can be turned on or off depending on environmental conditions. These mutations alter phenotype in one set of conditions, but not in another. A spontaneous mutation arises naturally due to cellular processes, such as errors during DNA replication. Induced mutations occur when DNA interacts with an external agent, mutagen, that causes DNA damage. The molecular basis of mutation. 1. Base substitutions. 2. Deletions and insertions. 1. Base substitutions. The simplest form of mutation is a base substitution, where one nucleotide pair in a DNA duplex is replaced by a different nucleotide pair. Some base substitutions involve replacing one pyrimidine with another pyrimidine, or one purine with another purine. These are called transition mutations. Other base substitutions involve replacing a pyrimidine with a purine, or a purine with a pyrimidine. These are called transversion mutations. In the human genome, the observed ratio of transitions to transversions is approximately 2 to 1. For example, in a T to C substitution, a thymine T is replaced with a cytosine C in one strand of DNA. This substitution temporarily results in the formation of a mismatched CA base pair. During the next round of replication, this transition mutation becomes permanently incorporated into the DNA sequence. The mismatch is resolved such that one daughter DNA molecule contains a GC base pair, while the other daughter DNA molecule contains an AT base pair. Transitions and transversions can give rise to different kinds of mutations, including A. Silent mutations.B, Missense mutations.C, nonsense mutations. A. Silent mutations. Silent mutations, also called synonymous mutations, occur when the nucleotide sequence changes, but the amino acid sequence remains unchanged. These mutations frequently arise from alterations in the third position of a codon, where redundancy in the genetic code often masks the effect. For example, a mutation that converts the codon AAA into AAG is considered silent because both codons specify the same amino acid, lysine. Silent mutations are not restricted to coding regions. Changes in nucleotides that occur outside coding regions can also be silent if they do not affect gene expression or protein function. B. Missense mutations. Missense mutations, also known as non-synonymous mutations, occur when nucleotide substitutions in protein coding regions lead to a change in the amino acid sequence of the protein. Such mutations can have significant effects on protein structure and function, depending on the nature and position of the amino acid substitution. A classic example of the phenotypic impact of a single amino acid change is sickle cell anemia. In this case, a mutation in the beta-globin gene changes the DNA sequence from GAG to GTG. As a result, the codon specifies valine instead of glutamic acid, leading to the production of sickle cell hemoglobin. C. Nonsense mutations. A nonsense mutation occurs when a nucleotide substitution creates a new stop codon within the coding sequence of a gene. These mutations lead to premature termination of protein synthesis, producing an incomplete polypeptide. The truncated polypeptide fragment is almost always non-functional due to the loss of critical amino acid sequences. An example is found in the beta-globin gene, where a mutation changes the codon AAC to TAC at the 17th position. This alteration introduces a premature stop codon, resulting in a polypeptide chain only 16 amino acids long. Such nonsense mutations are among the genetic changes associated with the disease β-thalassemia. 2. Insertions or deletions. Insertions or deletions of nucleotides can also occur in DNA, though their frequency is considerably lower than that of nucleotide substitutions. A classic example of the phenotypic effect of a small deletion is the human hereditary disease cystic fibrosis. This disorder arises from a defect in the gene encoding the cystic fibrosis transmembrane conductance regulator. The cystic fibrosis transmembrane conductance regulator protein functions as a CAMP-regulated chloride channel that maintains mucus-free airways in the lungs. In about 70% of cystic fibrosis cases, a specific deletion of 3 base pairs occurs in the cystic fibrosis transmembrane conductance regulator gene. This mutation eliminates the codon for the amino acid phenylalanine at position 508, delta F 508 mutation. When the length of an insertion or deletion is not an exact multiple of 3 nucleotides, the mutation disrupts the normal triplet reading frame of ribosomes. As a result, all amino acids downstream of the mutation site are altered, producing a completely different sequence. Such mutations are called frameshift mutations because they shift the reading frame of codons in the mRNA. General classes of DNA damage. Mutagen. A mutagen is any chemical or physical agent that increases the rate of mutation above the natural spontaneous background level. Spontaneous DNA damage can occur naturally, often through the action of water in the cell's aqueous environment. In 1927, Hermann Müller first demonstrated that X-rays are mutagenic in Drosophila. Since then, a wide range of physical agents and chemical agents have been shown to increase mutation rates by damaging DNA. DNA damage refers to any alteration that introduces a deviation from the normal double helical structure of DNA. The major classes of DNA damage are single base changes, structural distortions, DNA backbone damage. 1. Single base changes. A single base change, also called a conversion, alters the DNA sequence, but usually has only a minor effect on the overall DNA structure. An example is the replacement of the amino group of cytosine with oxygen, which converts cytosine into uracil, a base normally present only in RNA. This process is called deamination. Deamination is the most frequent and significant type of hydrolytic damage. It can occur spontaneously due to the action of water or be induced by chemical mutagens. When a UG base pair replaces a CG base pair, the DNA double helix undergoes only a slight structural distortion. Although this type of damage does not usually block replication or transcription, it can still result in the production of a mutant RNA or protein product. Vertebrate DNA often contains 5-methylcytosine instead of cytosine. Methylated cytosines are hotspots for spontaneous mutations because deamination of 5-methylcytosine produces thymine. As a result, a GC base pair can be converted into an AT base pair when the damaged DNA undergoes replication. Alkylating agents, such as nitrosamines, can cause the formation of O6-methylguanine. This modified base frequently mispairs with thymine, producing a GC to AT transition when the damaged DNA is replicated. Oxidizing agents, generated by ionizing radiation and free radical-producing chemicals, can damage DNA bases. Reactive oxygen species, O2-, H2O2, and OH-, can generate 8-oxoguanine, OXO-G, a guanine derivative with an extra oxygen atom. OXO-G is highly mutagenic because it can mispair with adenine, leading to a GC to TA transversion. This type of mutation is among the most common mutations found in human cancers. 2. Structural distortion of DNA, ultraviolet, UV light, has harmful effects on cells due to its selective absorption by DNA. DNA bases strongly absorb radiation at a wavelength of about 260 nanometers. The most frequent UV-induced DNA lesions are the formation of pyrimidine dimers, especially between 2 adjacent thymine bases. These dimers are also called cyclobutane pyrimidine dimers because a cyclobutane ring is formed by covalent bonds between the carbon atoms 5 and 6 of neighboring thymines. Since these covalent bonds occur between thymines on the same DNA strand, the normal hydrogen bonding with complementary bases is disrupted. This leads to distortion of the DNA double helix. Such structural distortion can block the progression of DNA and RNA polymerases, thereby impeding both replication and transcription. The formation of pyrimidine dimers represents a more severe defect compared to single-base changes. UV irradiation can also produce cytosine-thymine dimers, known as pyrimidine 6,4 pyrimidone photoproducts. Other bulky DNA adducts can be caused by chemical mutagens, such as large polycyclic hydrocarbons or alkylating agents. Intercalating agents, e.g., ethidium bromide, also cause DNA damage. These compounds contain flat polycyclic rings that insert between DNA bases, stacking with them in the double helix. This distortion of the helix can lead to the insertion or deletion of base pairs during replication. Base analogs can also induce structural distortion. These are compounds similar in structure to normal DNA bases, allowing them to be taken up by cells, converted into deoxynucleotides, and incorporated into DNA during replication. However, because of subtle structural differences, base analogs mispair inappropriately. For example, 5-bromoracil, an analog of thymine, can mispair with guanine, leading to mutations. 3. DNA backbone damage. DNA backbone damage includes two major forms. 1. Abasic sites, loss of the nitrogenous base from a nucleotide. 2. Double-strand DNA breaks. A. Abasic sites. Abasic sites are generated spontaneously, often due to the formation of unstable base adducts. In purine nucleotides, the sugar-purine bonds are relatively unstable and prone to hydrolysis. Hydrolysis of the N-glycosyl linkage in a purine base by the action of water results in the base being lost, leaving behind a hydroxyl group -OH in its place within the depurinated DNA strand. B. Double-strand breaks. Double-strand DNA breaks can be induced by ionizing radiation, e.g., X-rays, radioactive materials, as well as a wide variety of chemical compounds. These breaks are considered the most severe type of DNA damage because both DNA strands are disrupted simultaneously. If not repaired properly, double-strand breaks can lead to genomic instability, chromosomal rearrangements, or cell death. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!